behalf of Adelaide Oncology, I would like to thank Dr. M.B. Agarwal for giving us this opportunity to be a part of this webinar. Now I welcome Dr. Shashi Bansal. She is the chief guest of this program, and Dr. Neeraj Arora, our guest speaker. With your permission, I would like to give you a small brief about the Adelaide Oncology. It is one of the fastest growing oncology company in India. It is having more than 125 products in its kitty. Not only this, it it is accredited with many uh, international bodies like Anvisa, Invima, PICS, and EAEU approval. It is having global footprint in more than 46 countries. Adelaide Oncology is having a strong portfolio in hematology basket. It started with the dasatinib. Dasatinib, we have launched the first generic brand, Bidan, for dasatinib. Then we have launched many other hematology products like carfilzomib, eltrambopac, then nilotinib. Recently, we have launched pomalidomide. We have launched the most affordable brand, acalabrutinib. Then we are going to launch many NDDS formulations for the patient benefits. Adelaide Oncology has a commitment to provide drugs at very affordable price and high quality products. That's the reason Adelaide Oncology has reduced the MRP of many of its oral products to pass on the benefits to the patients. In our core, patient first is our approach. With this approach, we have a strong R&D which is providing the NDDS formulations. We provide the best of the best patient support to the patients on the discretion of the oncologists. Not only this, we try to disseminate the knowledge and provide most affordable medicine to the patients. So with this, I would like to thank Dr. M.B. Agarwal sir once again and hand over the session to him.
Thank you, Prashant, for setting the ball rolling. Also, thank you for hosting me last week at Panchkula. It was wonderful to see your office and enjoy the afternoon with you and your group.
Thank you very much, sir.
I'll just share my screen. So, good morning to everybody from India, and good afternoon, good evening for those who are logged in from the rest of the world. Today, our guest speaker is Dr. Neeraj Arora, who has spoken to us multiple times in past as well through these webinars, as well as in person for various MHG midterm and annual meetings. Today, he's going to talk on the basic aspects of molecular hematology for the non-molecular hematologists. That is basically to the clinicians, chiefly the students, and all other colleagues. We have invited undergraduate, postgraduate, DM and DNB hematology and medical oncology students to join today as delegates. And we also kept a quiz exam for them after 15 days with very attractive prizes for all the three groups. And let us see how much they absorb from this lecture and benefit. This webinar is brought to you by Mumbai Hematology Group, supported by Adelaide and managed by Perfect Square. I thank Mr. Ashutosh Shukla and Mr. Prashant Agrawal from Adelaide, Mr. Yash Kalpesh and their team from Perfect Square, the Executive Committee of Mumbai Hematology Group, and most importantly, our chief guest, Dr. Shashi Bansal from Jaipur, and our guest speaker, Dr. Neeraj Arora from Ahmedabad. Of course, I'm indebted to all the discussants, hematologists, hemato-oncologists, and molecular hematopathologists who have joined as discussant today, and all of you participants from various parts of the world who have joined on this Sunday morning, afternoon, or evening to be a part of this very important webinar. To give you a flavor of the next week academic activities of MHG, Saturday evening, 7 o'clock IST and 8:30 a.m. Central Time US, we have Dr. Meera Mohan from Wisconsin speaking to us about the bispecific antibodies in the management of relapsed refractory multiple myeloma. Next Sunday morning, that is 6th April at 11:30 a.m. IST and 8 a.m. Italian time, where Dr. Maria Capellini from Milan speaking to us on an unusual problem, that is problems of thalassemia major after the age of 30 years. We have a galaxy of discussions today. I will not be able to introduce all of them, but I have them placed here alphabetically in this and the next slide. Now it's turn to introduce our guest speaker for the day. He doesn't require any introduction to the audience from India and probably even from those who are outside. As I mentioned, he has taught us about the molecular hematopathology at a number of occasions during last 5 years since the beginning of pandemic. He's MD pathology, PDF hematopathology, PDF molecular hematology. At present, he is director of the molecular biology at the prestigious Unipeth Specialty Lab at Ahmedabad. He is a molecular pathologist par excellence. He gave his services for 7 years to Tata Medical Center, Kolkata. Prior to that, he worked in the Department of Hematology, CMC, Vellore. He is instrumental in setting up affordable diagnostic molecular techniques for both hematological and solid tumors, as well as benign disorders, including the techniques like NGS, of which he is a master, and the digital PCR. Over and above this brief introduction, you will always see him smiling, not only in this photograph, but also in person. I've never seen him when there is not a smile on his face. So that's wonderful, Dr. Arora. As I mentioned, he will be lecturing on the basics of molecular hematology for us. Before he does that, let me invite our special guest. And today that is none other than Dr. Shashi Bansal from Jaipur. Once again, all of you know her. She's senior consultant, hematopathology at the prestigious Bhagwan Mahavir Cancer Hospital Research Center, Jaipur. Just last week, Bhagwan Medical College and the hospital had a big CME in hematology at Hotel Radisson Blu, and I had the opportunity to be there for that event. She did her MBBS from Maulana Azad Medical College, New Delhi in 2002. This was followed by MD from Lady Hardinge Medical College, Delhi in 2006. She also did her DNB, the National Board exams, next year, that is in 2007. She has vast experience in hematopathology, and this includes flow cytometry, bone marrow aspiration, trophine biopsies, lymph node histology, especially for hematolymphoid malignancies, including immunohistochemistry. Her main areas of interest are flow cytometry and IHC for hematolymphoid malignancies. She's associated with teaching under NBE for last 11 years. She is guide and co-guide for DNB and PDCC students. She has been examiner at various occasions. She has many presentations in national and international conferences and publications in various peer-reviewed journals. She has organized various hematologic CMEs, updates, workshops, and quiz competitions. She received the Best Oral Paper Presentation Award by Delhi Society of Hematology way back in 2008. She is presently the Joint Secretary of Rajasthan chapter of IAPM. She is life member of IAPM, ISHBT, TCS, PPSRAJ, and an active participant as well as faculty member of various CMEs workshops and hematology conferences. We are privileged to have her present today to inaugurate this webinar and give some words of wisdom to our colleagues and students. Thank you, and over to Shashi.
Very good morning to everyone. It's a great pleasure to be here today and to inaugurate this insightful, important session on molecular hematology. First, I want to thank the Mumbai Group of Hematology and Dr. MB Agarwal sir for organizing this wonderful event and for their continuous effort in spreading the knowledge and awareness in field of hematology and developing a strong community of learners. These kind of online activities decrease the gap in between the pupils residing in the metro city who have the easy accessibility with the advancement, and the remote area by providing the new advance in medicine at their own places. Sometimes we are busy, we don't have the time to go and attend the sessions, but these online activities provide the platform to learn from these things. To all the students and young doctors and professionals attending today, this is an exciting time to be in the hematology. The medicine is advancing faster than ever, and molecular science is helping us understand disease in a way that once impossible. We are now able to detect the disorder earlier, treat them more effectively, and even personalize treatment for the better patient outcome. This is just not the progression, it is a revolution. Each of us has the opportunity to be the part of this revolution. But while the science and technology are advancing, one thing remains the same: the role of dedicated, passionate, and the curious doctor with the perfect work and family life balance. The knowledge alone is not enough. What truly makes a great doctor is ability to think critically, to keep learning, to stay committed to the patient care with a calm mind. Because human nature has to be understood. Every discovery, every innovation begins with the simple question: how can we do better at our place? And it is that mindset, the willingness to seek the better solution that lead to the progress. We all are not residing in the metro cities or the big cities, but each of us are important in our own place in the patient care. I work in a cancer hospital by always respect and give appreciation to the person who first clinically suspect or diagnose the patient at the remote area with the minimal facility or the morphology alone, and refer the patient with the appropriate higher center at the correct time so that he is saving a life. So I know that the journey of the medical education is long and sometimes very overwhelming. Late nights, endless studying, constant pressure, it can be exhausting. The effort you put is today will impact the life of you or your patient. Tomorrow, your skill and knowledge will bring hope, relief, and healing to those who is in need. There is no greater reward than that. So medicine is always evolving, and the best doctors are those who never stop growing. So keep asking questions, challenge idea, and most importantly, stay passionate about what you do, whatever level you are, and whatever you're doing. Every person is important is in this healthcare profession. So I encourage all of you to embark the learning, not just as a requirement, but as a lifelong habit. Habit does not goes. So it keep on learning. Today is also the Gudi Parva and the first day of Chaith Navratri, which symbolize with the strength, endurance, and selfless service, the quality that resonate deeply with the healthcare professional, who are the modern day warrior in the fight against the illness and the suffering. With that, I honor to declare this webinar open. And today, I hope the session will give you the valuable insight and inspire you to keep pushing forward further. With that, I thank you and best wishes to all. Thank you, sir, and thank you, everyone.
Thank you, Dr. Shashi Bansal, for that beautifully crafted and great speech full of knowledge and advice. Before I invite Neeraj to give his wonderful and masterly talk on a very, very important subject, because there are a large number of students listening to you today, I must tell them one thing. The pathologist or the diagnostic person very often works behind the curtain. The clinician treats the patient and gets the credit for helping the patient, sometimes curing the patient. And I say sometimes because we are dealing with a difficult branch all the time. Whatever credit we get, the brain behind that is the one who's going to speak to you today. There is nothing better than a correct diagnosis in the field of medicine in general, oncology especially, and most importantly, hemato-oncology. What you're going to listen today gives us an accurate diagnosis at the baseline. It sort of splits up a broad diagnosis from leukemia to acute myeloblastic leukemia to its genetic subtype. Not only that, it guides us how to fine-tune the treatment of one person from the other person. It allows us to give the prognosis to the patient. You are not going to survive more than few weeks, or you are going to be cured. are two very different prognoses, the basis of which is what Neeraj does for us. Not only that, afterwards, when we start the treatment, we come back to Neeraj for probably telling us whether we have achieved what we want to achieve. These are days of targeted therapies, and we want that target to be totally destroyed. Whether that target has been destroyed or not, he is again instrumental in telling us. These are days of MRD. In most of the diseases in hematology and hemato-oncology, we are looking for achieving non-measurable residual disease. Once again, Neeraj tells us whether that has been achieved or not. It's not important only to give happiness to the patient, it also helps us in stopping the treatment, saving all the toxicities of the drugs, saving a lot of money, and probably helping not only the patient, not only the family, but probably the country. So importance of an accurate diagnosis at the molecular level is so immense, which at the age of 70 plus, probably I'm still learning. So Neeraj, I'm going to be your most important student. Please go ahead.
So, good morning to all of you, and thank you so much, sir, for those kind words. It has always been an honor and pleasure to be here. We have always been learning from you, the way you handle, the way you do things, and always setting up the benchmark higher in every way that you do. And very well said, Dr. Shashi, that we keep learning from each other. So for the today's session, the topic that was asked for me was ABC of molecular hematology for the clinicians. So the target audience here is the clinician who has never visited the laboratory, or the people who have not visited the laboratory, to give you a sense of the things happening in the lab. And the basic idea behind this is to give, so that you are able to interpret or understand the report coming out, or the technical aspects coming out, because many a times we just see it as a positive, negative, and don't have a sense of the what is the things happening behind it. So that is the intent with which I'll be taking this forward. And the my talk is structured in a way like in that first there will be an overview of basics, and then I will be touching upon PCR, then real-time PCR, something on digital PCR, Sanger sequencing, some of the basic things that we all should be aware, and then a few things about next generation sequencing. It is not possible to be able to touch upon everything in this, but the idea is to be able to give you a snapshot of the things, the issues or the concerns that which all we must be aware. So as we start now, so we all know our body or a tumor is made up of cells, and within those cells we have chromosomes, and from those chromosomes, within those chromosomes you have some DNA, or at a genetic level the functional unit of this, that is we call a gene. So we have a cell, chromosomes, and DNA, or a gene. And typically in the assays or techniques like cytogenetics, we look up at the resolution of chromosomes through techniques like karyotype and FISH. We look up at a higher resolution in cytogenetics. And when we talk of molecular, we go down slightly more depth in terms, we deal with DNA, RNA, and we have techniques like sequencing coming into this picture. And when we all must be aware, we have already read that, so down to the, so we have cell, chromosomes, gene, and the structure of the gene. And if you look at the structure of this gene, we have DNA, where we have a promoter region. You have exons and introns. There could be different number of exons or introns in a particular gene. like for example, CEBPA, which is a single exon gene. So there is a single exon, but very difficult to amplify and proceed for downstream applications. But the overall structure of all the genes remains same. That is promoter region. There are exons, introns, and through the process of transcription, where the introns are spliced out, we got something called as RNA. And through the process of translational, we go down to the protein level. So we have cells, chromosomes, gene. Then we have within these genes, the DNA, RNA, and protein. That is the central dogma. And any process or anything interrupting at a DNA level interrupts the RNA and subsequently causes the dysfunction of the protein. So the protein which was functioning normal way, causing its normal function, is disrupted, and hence leads to the issues that we see in different diseases or different subsets. Now, we have all known how this works out. Now, this is another way of looking at the same things. So whatever I mentioned, like central dogma, DNA, RNA, and protein, this is how a lab looks at the DNA, RNA, protein. So this is DNA. So whatever shown in the left side is the DNA. You have something in the red. There all are ATGCs, and we know this is exon 1. Then you have intron 1. Then exon 2. And then you have intron. And when you splice out the ATGCs in the intronic regions, we have what is, we call it as a RNA, and this is how RNA looks like. And we have read the coding regions, like in our biochemistry classes, we know that ATG codes for methionine, and this leads to the protein function. So this is a same laboratory aspect. So giving you an overview about how we see in the laboratory, everything is ATGC, but we know by sequencing or by the how this structure is, and how do we know that this is exon 1 and intron 1? By comparing with the literature, or this data is already available. So we go down to the reference sequencing, download the sequence, compare that with the reference sequence, and then we are able to see that whether this is same or there is any change in these variations. Like ATGC would, so there is a triplet codon. So this would be codon 1, codon 2. Subsequently, like that, you have codon 1s, like V600E, point mutation at codon 600 position, valine is changed to the other amino acid. And that is how we understand the various changes or mutations there. But the basic structure is DNA, RNA, and protein. And this is the laboratory, how it looks like. Now, what is a change or a mutation? So this mutation means a mutation means a change in any change in this genetic material, which is a single base or a large segment of a chromosome. Any change is there, and if that change is present at a frequency of more than 1% in the population, we tend to call it a mutation. We must remember that there are changes, normal variations between two people, and those are called as polymorphisms. We all have the same nucleic material, but we all are different because of this normal polymorphisms or variations which are there. And we need to distinguish this by from the pathogenic variations or from the harmful mutations or variations which are there. And some of them are known, some of them are not known. And this is what a problem comes when we are describing in terms of hereditary cancers or germline mutations or variations. Like MTHFR, these are polymorphisms, these are not mutations. But TP53 pathogenic variations or BRAF V600s, these are mutations. So we keep evolving, we keep learning and learning about those variations which we get during our course. So as we sequence more and more, we keep getting those and understanding those changes. What are the types of variations which are there? there could be various types of changes or deletions, or it could be a substitution, a point mutation changing to another. It could be a deletion, it could be an insertion, and this could be divided into purine to purine or pyrimidine to purine, a transition or a transversion. Though in clinical sense, it doesn't make too much of a difference. It is this doesn't have too much clinical significance, but we should be aware that. So these point mutations could either be a missense mutation, where because of the change at one position, T2A change leading to change in the amino acid. So from a normal amino acid, we have now another amino acid which leads to disformed protein and causing affection of the protein, or a nonsense, where because of one point mutation, there is a stop codon. So the protein cannot grow further here. So it leads to abrupt short protein which functions again abnormally. It could be a frame shift. There is an addition of a single nucleotide leading to the entire shift of the frame, and there could be either a deletion. So these are different types of point mutations or deletions that we could see. Now, so we just saw about mutations, changes, or polymorphisms which affect and how we look at them in the laboratory. There are two aspects. One is germline mutations and somatic mutations. So germline means whichever is present in our genes, and somatic means anything which is related to the tumor. So tumor means we have to have any sample that we are using, it has to have the blast percentage, blasts, or an FFP specimen from a tumor for characterization of those somatic mutations. It cannot happen that we are using a peripheral blood sample and we expect to pick up the somatic mutations. So for somatic mutations, we need tumor. For germline, we need EDTA, buccal swab, or a skin biopsy specimen for better characterization. So the sample typing is very important as we go forward, or as we learn to see whether this is a germline or somatic. So what are the common techniques we use for molecular diagnostics, and how to pick up a particular technique for a particular disease? This would depend upon the target disease. So what disease we are targeting? Typically, when we are looking for fusions, a FISH, karyotyping on a RT-PCR, and even now an NGS, or which is RNA-seq or a whole genome or a targeted sequencing is able to pick up. But depending upon the need, I want to pick up within a day. So then I might want to use a FISH as opposed to other techniques. I want to quantify it, then I might want to use a quantitative real-time PCR assay. I want to do hundreds of fusions in one go, then I might want to use an NGS. I want to pick up entire spectrum of any fusion which is there, then RNA-seq or a whole genome approach could be there. So depending upon my need, I want to pick up the technique for that. Similarly, for copy number alterations, FISH, karyotyping with low resolution, MLPA are common techniques, but we are all moving down to NGS in all these areas. And now even techniques like optical genome mapping are there. Typically for single gene alterations like BRAF, V600E, L265P, there are allele-specific PCRs. We want to target a specific point mutation or a real-time PCR which would fit JAK2 V617F. So real-time allele-specific PCR are two good approaches to be able to pick up. We don't use Sanger sequencing, or not encouraged to use Sanger sequencing in these scenarios because of the sensitivity of this assays. And now, since everything is moving into an NGS space, we are looking more and more towards NGS. And the typical sensitivity of these assays, real-time PCR, is around 1 to 2% sensitivity. You have Sanger sequencing with the sensitivity of around 20%. You have different PCRs with different sensitivities. NGS would have a sensitivity of 1 to 5%. So depending upon the sensitivity and my need and requirement and my disease type, I am able to pick up the technique and use that for my clinical detection. This is, as I mentioned, for larger deletions, where I want to pick up more than 60 or more than 100 base pair, or a whole exon level deletion, or multiple exon level deletions, MLPA is the technology of choice. This is one graph to show the alpha multiple MLPA, where this shows the deletions. Typically, for thalassemias, you have beta and alpha thalassemia, and you need to, for alpha thalassemias, there are a lot of deletions, 3.7 kb deletion, 4.2 kb deletion, and then fill del, a lot of deletions are there. So you use MLPA, and this shows a deletion over here. Also, it helps you pick up multiplication. And this is one MLPA graph showing alpha multiplication. Now, a scenario, I just wanted to bring that scenario, like in thalassemia, when the presentation is thal inter, and you are able to detect only a point mutation in beta globin gene, and you're not able to detect other mutation, or you're not able to get the second mutation. So in those scenarios, sometimes alpha multiplication gives rise to the thal inter-like phenotype. So a single point mutation in beta with an alpha multiplication can give rise to a thal inter, and this technology is used to be able to pick up. So MLPA to be able to pick up larger deletions. Even now you have a digital MLPA being considered. So combining the power of MLPA with NGS, and this is now being used in hereditary cancers, where I can look at deletions of larger number of genes, maybe 20 genes, 30 genes. And in cases like myeloma, and in cases like ALL, where this is being explored and available. But now MLPA in hematology, typically for thalassemias. Now, so we learned about MLP and other technologies which are there, but on the background of all the techniques, the one assay which forms the backbone of every hematologist or hematopathologist or molecular pathologist is a PCR. So what is this PCR? You must have heard this. So PCR is just amplification of your region of interest, amplification of your DNA of interest. And this has not been described too long ago. It is described only in 1989, but it has been so part of our lives. We cannot think life beyond PCR. So PCR is simple amplification of my region of interest. How do we do it? It has got more refined now. There are thermal cyclers available. These are called endpoint thermal cyclers. There are different variety, and we use this to amplify my region of interest. And when I want to amplify, I program that thermal cycler to be able to multiply it exponentially. So this is, we cycle around 30 to 35 cycles. And at the end of this PCR, we are able to get 2 raised to power 36 copies of my region of interest using primers of my region. It could be TP53, it could be BRAF, or it could be any other gene. And I look it into a gel. This is a gel showing my DNA or my amplified product. And this is how I look up in a PCR. So a simple amplification of my region of interest. There are a lot of factors which affect the PCR. So you must have heard this PCR is not working. So there is a problem in the PCR. You know, we are not able to amplify. One of the most important component here is the nucleic acid or the template. It could be DNA, it could be RNA, it could be peripheral blood DNA, RNA, it could be FFP DNA and RNA. And typically, FFP DNA and RNA are slightly difficult to amplify because of multiple factors: how the process, how we extract, and depending upon the paraffin, how it has been fixed. All of those factors affect the quality of the nucleic acid, and that is of paramount importance. Again, in an RNA setting, you must have heard this, that that you are not able to amplify, it has failed, or QC failed. So once a sample reaches to the laboratory, the first thing we extract the DNA and RNA and do a QC. And this QC is done using these three key equipments in the lab. One is a nano drop, very commonly used for routine PCRs. And then we have a qubit and an Agilent tape station, mainly used in the NGS settings. So we use this nano drop or qubit for quantification and quality. And we use the ratios 260 by 280 ratio. It should be between 1.8 to 2.0 to check that whether this is of good quality DNA or bad quality DNA. And also since we have started using liquid biopsy for solid tumors, and even now it is coming up for hematological malignancies as well. So where you have cell-free DNA, cell-free TNA, cell-free RNA. So all those things are there, mainly using the plasma. We extract the cell-free TNA and then check the quantity and quality using this. And these are most important steps of any PCR or any downstream things that you do. If this is not right or there is a problem in this assay, nothing downstream, no matter whatever we do later on, is going to work. So we have PCR, simple amplifications. Now we have a modifications of those PCRs. So different modifications of those PCRs is what we use in our clinical practice. It is endpoint PCR, as I mentioned. When we amplify, only in the end you are able to see that what is amplified or not. So using that endpoint. Long range PCR. Typically, this PCR is used in alpha thalassemias, where I have to amplify 3.7 kb deletion. So usually I am able to amplify 1000 base pairs. So a simple PCR would be able to amplify around 1000 base pairs. Anything beyond, I need a separate modification in the process, and then we call it long range, and we use it in thalassemias. Gap PCR, typically to pick up deletions. Again, in thalassemia settings, we use this gap PCR. So if I am able to amplify 1000 base pairs and there is a 600 base pair deletion is there, so I will get 400 base pair band. So that is how I call it a gap PCR. So depending upon the deletion, I will be able to pick up that gap. Fluorescent PCR. So you have two primers, and one of the primer is labeled to fluorophore and gives fluorescence. This fluorescence is picked up on a Sanger sequencing platform. This is a fluorescent PCR. It gives you more sensitivity to be able to. I'll show some examples. Multiplex PCR. Instead of 2 genes of interest, I amplify 4 or 5 genes of interest using multiplex PCR. Allele specific PCR is when I'm targeting a single point mutation. It is a T2C, JAK2V617F. These are allele specific PCRs. It could be a real-time or a gel-based allele specific PCR. So this is a sensitivity down to around 1 to 2%. Very sensitive PCRs. Nested PCR. When I have 2 rounds nested PCR, so round 1 PCR, the end product of round 1 is used as the template for the round 2. So you have 2 PCRs going parallelly, and this gives more sensitivity to whatever things I am doing. Reverse transcriptase PCR. So usually we start with the DNA, but when I'm using RNA, I convert it into a cDNA and then run my assay. So this is a reverse transcriptase PCR, real-time PCR. So endpoint is when I'm seeing my product in the end, but here I am monitoring that real time, and we all know real time PCR because of the COVID. Nobody is a stranger to the real time PCR. Then there are other modifications of hot start, inverse, and cold PCRs. So a lot of modifications of the same process but these are all amplifications and modifications. Some of you might invent few more PCRs because this is a very dynamic technique and you can modify to your interest and still be able to use it for your clinical testing. So few examples of this PCR. This is a commonly used NPM FLT3 ITD in AML, and ITDs are many a times are missed out in an NGS, and we always prescribe to check it by parallelly by simple gel-based or a fluorescent PCR. And this is an example. That is why I showed here. So on the left side is what you see is two genes of interest amplified. And on the right side, those genes are shown using a fluorescent PCR on a Sanger sequencer. And you can see here in the NPM, there is a wild type peak and there is a 173, is a 4 base pair insertion. So this NPM is a mutated NPM, there is a 4 base pair insertion. I am not able to tell what those insertions are. There are more than 35 different NPM mutations known. It could be TCGA, it could be TCCT. I am not able to tell that, but I am able to tell that this is mutated. Why this characterization is important? Because for MRD settings, for downstream quantification of this NPM, I need to know what is this type of mutation. So I need to Sanger sequence or NGS to be able to find out what is the exact mutation and then use that information for my further downstream other applications. Similarly, this is a FLT3 ITD assay. This is a wild type peak and this is a mutated ITD. So this is around 40 base pair ITDs being picked up in a multiplex fluorescent PCR. So this has a sensitivity down to around 1 to 5%. Anything below that, it is not possible. And this ITDs, a bigger ITDs, more than 180 or 200 base pair ITDs, detecting on an NGS is a problem. And so you always tend to do a parallel this PCR whenever you are doing an NGS for FLT3 ITD. So this is an example of a fluorescent PCR. Another example of a full fluorescent PCR is calreticulin in MPN, a wild type peak here and a mutated peak here. This is a 52 base pair insertion being picked up. So this is a type 1 calreticulin mutation. Just to show you a gel. So first we amplify, check whether it is amplified, and then do a fluorescent PCR and check it on the Sanger sequencer to see that this is mutated or not. Another example of a fluorescent PCR is a clonality assay, which we tend to do, so here you can see polyclonal peak. So polyclonal peak being shown here, and then you have a control, and this assay showing that there are two clear peaks showing this is clonal. Example of a fluorescent PCR using kit or primers which are there from the market, and then looking it into a Sanger sequencer. So another example is this is an allele specific PCR, which is a gel based allele specific PCR. So here is a positive control showing two bands. Here it is a negative control, and all these samples are negative. So a modification of an allele specific PCR. There are other modifications as well. So this is an MYD L265P, typically described in lymphoplasmacytic lymphomas. You have this patient which is showing negative. This is a wild type, this is a mutated, and this patient is actually showing two peaks. One is wild type and one is mutated. So one mutation, normal variation is picked here and the abnormal variation is picked here. So meaning this is a mutated L265P mutation. So that is some variations in the allele specific PCR and how we use that. JAK2 PCR is something separate. This is a separate, so slight modifications. We could also use a real-time PCR to be able to detect same things. This is a gel-based. I can use real-time around same sensitivity, but then using a quick within 2 hours, I should be able to detect that this is there or not. Nested PCR, again I mentioned this is round 1 PCR. We use a template for round 2. So typically using nested RT-PCR, we use for our qualitative detection of our fusion genes, giving more sensitivity to whatever we want to detect. Inverse PCR. So this is used in the regions where we don't know the sequence or what sequence we have to do. Typically, this is used for intronic inversion. So like for intron 22 inversion. So for hemophilia. In severe hemophilia, the commonest mutation is the intron 22, and detecting those inversions on routine NGS or other assays is difficult. Either we use a long range PCR or we use an inverse PCR, and we have set up a inverse PCR for intron 22, and this is also shown in intron 1. So basically all these are modifications of the PCR, different modifications in different way. You amplify your regions of interest and we are able to look down. Again, it is important to know what is the sensitivity of those assays, those gel-based assays. Typically, gel-based assays are less sensitive and real-time PCR-based assays are more sensitive. So real-time is, we are able to reliably detect our regions of interest down to the sensitivity of 1 to 2%, and we require separate equipments. These are some of the equipments which are available in the market: QuantStudio 5, QuantStudio 7 from Thermo Fisher. Then there are other assays like BioRed, PenDoff. Different real-time assays are available. But the basic principle in the real time is there is a primer and a probe, and on those probes there is a fluorophore and a quencher, so that there is no fluorescence. So this quencher kind of inhibits this fluorophore, whatever fluorophore is bound here, and there are different combinations which are available. So as we do a PCR and this primer synthesizes or sequences it, the fluorophore is released from this probe and it is emitted into the reagents, and this we measure it through different technologies. We measure this fluorescence, and this is proportional to the number of cycles that the amplification is taking place. So in the endpoint, only the DNA was amplifying, but here the fluorescence or the fluorophore is amplifying in proportion to the cycles that we have. And we measure this fluorescence intensity as this the fluorophore is released into the reagents, and the cycle at which this happens is what we call the CT. So based on, COVID, we all know what is the CT value. So basically CT is the threshold cycle. So we have programmed this real-time PCR to amplify my regions of interest, and this fluorophore is increasing based on the number of cycles, and the cycle at which this fluorescence crosses the threshold is we called as CT. The more the amount of material which we started, less is the CT, less the material here, more is the CT. And this is what we use for our all real-time PCR assays. So if my region of interest is very small, my CT is very high. If my region of the whatever I'm targeting is more, then CT will be less. One example of a real-time PCR is a quantitative BCR-ABL. So this is the flag bearer of molecular hematology, where we have real-time quantitative assay being utilized in the laboratory, and we need to be aware what happens in the lab in this. So this is a plate which goes into this machine that I showed, and what we design, this is the design of that plate. So each of this plate will have different components. So basically here we have ABL control gene and a fusion gene what we want to target. It could be BCR-ABL, it could be PML-RARA or other fusions. So we have standards shown here in duplicate: 10⁶, 5, 4, 3, 2, 1. Based on this standard is what we are quantifying my template or my sample. Similarly, from the fusion standards, we are able to derive the value for the sample, again put in duplicate or triplicate. And this is just to show that how does a template look like. So there are control gene standards, there are fusion gene standards, and there are unknown samples which are there, again in duplicate or triplicate, and we derive those values from this standard curves and get those values of BCR-ABL, the mean ABL copies, and get a ratio for the BCR-ABL. Using a real-time PCR is what we tend to do that. this is on deck. On deck, how does that standard curve look like? Same, whatever I showed, that was plate. And this is a software view showing that this is in red is the ABL standards, and on green is the BCR ABL standards and samples. And these are the standard curves. Based on these standard curves, we derive the value for each of that gene, and then divide them and get the percentage values for the BCR ABL. So application of real-time PCR moving forward, we have now a lot more digital PCRs coming into the space of rare event analysis. Real-time PCR in quantitative assays is the backbone for all quantitative assays, and very well practiced because of this scale, availability, reliability, guidelines availability. Real-time PCR is still the gold standard, but we have digital PCRs offering or coming into this space into rare event analysis. It could be liquid biopsy based testing. It could be BCR-ABL quantification through digital PCR, even some other quantitative assays as well. And here you have digital PCR / droplet digital PCRs. So what we do here is you divide either through digitally, or what we call it as a chip-based division. So you have divisions which is chip-based or a droplet digital base. So droplet digital is typically associated with Bio-Rad, and there are other companies available. And you amplify again. So basic principle remain the same. You are able to extract, then you do divide your region of interest. It could be chip-based division or a droplet-based, and these droplets act as PCR, meaning they are able to amplify everything, whatever is in that. And now you have the droplets or the divisions where some of them are having those regions or those fusions or mutations which we want to target or which we want to quantify, and there are some droplets which we don't have that. And then using a Poiseuille's equation and a calculation, we are able to derive those values based on the whatever droplets or divisions are showing positive, and based on whatever divisions are not showing those divisions. So based on the number of positive droplets and the calculation is there, so we are able to use and get an absolute value for whatever we are measuring. Why it is important? Because it gives you absolute quantification. There is no need for standard curve, and this is very, very high accuracy of precision and reproducibility. Whatever value comes in my lab, it is very reproducible in other lab versus other country, and this has been shown that this is very, very reproducible. So that is why in rare event analysis, in MRD settings, in this PCR is kind of the place where everyone is looking at. We will have more usage of ddPCRs in the coming times. Already we have started seeing some of its uses. So there are many instruments available. One is the, as I mentioned, the Bio-Rad based droplet digital PCR, where the assays for BCR-ABL and other liquid biopsy settings are available, and you have a chip based digital PCRs, but the droplet digital kind of has a more market share, or they are more commonly there in the market, and more data and more people are using it. How does it look like in the when you do the test? This is one example of a this is one example where of a positive for BCR-ABL. You can see a lot of droplets here, and this is a threshold, so showing this is a positive for BCR-ABL, and again this is a negative for BCR-ABL based on this threshold. So here you are able to see one droplet, two droplets as well, and giving you a much more better sensitivity to be able to pick up. Yes, you need to be a little careful because 2-3 droplets, calling them positive, contamination or issues are there. That lab has to work out what lower values you want to report. So up till now, I have taken you through, the different basics like various mutations, fusions, how to pick up a what are the common technologies, their sensitivities, then the PCRs which are there, different types of PCRs basically which are amplifications. There are modifications of those PCRs which we use in our laboratory. Now moving further, we are moving to sequencing. So sequencing means the process of getting right, getting the nucleotide bases right is what we want to look at. So here there are different technologies are available. There is a Sanger sequencing, which is a gold standard sort of technology available. Then we had pyro sequencer coming into the picture, and now everything is NGS. A lot of NGS is being there. So, but I'll take you through this Sanger sequencing, as I mentioned earlier as well. It is a gold standard, and the sensitivity is around 20%. So if a mutation is there at around 10% level, then you may not be able to pick up using a Sanger sequencer, but you might be able to pick up this mutation using other technologies like pyro sequencer or an NGS or a real-time platform. So you need to be aware that Sanger sequencing is not the go-to choice for now for somatic mutation detection, and you need other technologies in this area. Pyro sequencer typically very good at works for FFP specimens for methylation settings, single gene mutation detection. So, but everything is moving into an NGS, and I'll talk of this as well. So how do we do Sanger sequencing? So there are systems available. This is how the Sanger sequencing is seen. This is a 4 capillary. There are 4, 8, 24, 26, 96 capillary. So meaning I can sequence 96 reactions in one go. So usually 4 to 8 reactions is what we require. And the output that we get using the software, there are different softwares available. This is how it looks like. So basically, in a Sanger sequencing, the heart of this Sanger sequencing processing is a Sanger sequencing reaction, which we do in the lab, where we pull in the regions of interest using amplification of that, then pull in regular nucleotides and dideoxynucleotides. These are labeled and these are terminators. So this is based on a chain termination method. So Sanger sequencing is a dideoxy chain termination method. So wherever this dideoxynucleotide will be lost, that chain will stop there. So basically we pull everything. As I mentioned, Sanger sequencing reaction is the heart, where we a lot of reagents will pull in, and you get fluorescently labeled DNA sequencing. Wherever this fluorescent di-NTPs are lost, the reaction stops there, and now you have different DNA sequences which have terminators. And as they pass through the laser, they give certain peaks based on the fluorophore, and this is processed through a software, and we are able to analyze the data. So this is a Sanger sequencing. This is a raw data looks like. So a lot of peaks coming into those things, and we now analyze through the software, and this is a processed software showing a Sanger electrophotogram. This is compared to the reference sequence which we download from various databases, and then we know about those sequencing, what is coding what protein it is coding, what the amino acid it is coding, where it starts, where it ends. We know that. So then we compare with the reference sequence, and now I know where is a change, and comparing that different types of changes we detect. It could be a deletion, it could be a point mutation, it could be a missense mutation, as I mentioned. So the Sanger sequencing good part, it is quick, lowest error rate. It is gold standard for a long time. The problem is the sensitivity. If we want to do whole genome, it will take tens of years. So I cannot use them for when I'm doing multiple areas. The NGS is the technology which helps me do that. But Sanger sequencing nowadays typically used in germline settings, single gene confirmation settings, where I already know the mutation I want to confirm, then I use a Sanger sequencer. Typically for somatic settings, because of the sensitivity of around 20%, we don't tend to use, not encouraged to use, though still being used in some of the assays. But that is Sanger sequencing. Now moving further, so one point here is also there that This instrument has another software called Gene Mapper. We call it gene scanning assay. Some of the fluorescent PCRs or some of the clonality assays I showed, the fluorescent measuring things are used using this instrument and that software, Gene Mapper software. So we use that for our multiplex fluorescent PCRs. We require this instrument and the software to be able to process that. I showed initial examples of that in my use, and we commonly use for NPM FLIT3. We use it for our clonality based assays. We use it for our chimerism based assays. So we use that. As I mentioned, now everything is moving into the line of next generation sequencing. So you are able to process multiple genes, multiple things in one go. This is massively parallel sequencing, and Illumina is the market leader here. There are a lot of instruments available based on your choice or what you want to do. You have to pick your equipment accordingly. There are other technologies also available. So, there is a Thermo Fisher and Illumina. So two technologies available. These act on the principle of mini pH meter. So each of the hydrogen ion which is released is detected, and that is how. So the principles are different in each of them, but the workflow wise, more or less they work on the same principles, that you are able to generate a lot of I mean, you use them into different assays. So how do you want to use those? And even now more technologies are also coming into this space. So these are, what I mentioned is, these are currently practiced available short read sequencers, where I divide my DNA into 150 base pairs, and then what I'm using is that. Now there are long read sequencers like PacBio or Oxford Nanopore coming into, but they're not into the clinic as yet. But going forward, we might be using more long read sequencing as also. So using these instruments, using these technologies, how we want to use it for my clinical sense, different ways in which I want to use. One is the whole genome. So whole 22,000 genes are sequenced in one go. All introns and exons are sequenced here. Exome sequencing, I am sequencing only the exonic regions, no introns here. So if you want to use your exome sequencing to pick up intronic thalassemias, that is not possible. Or I mean, that is not the technique. So we sometimes tend to think that exomes will be able to. So exome is not the technique to be able to pick up the intronic changes. Then targeted regions of 50 gene, 100 gene, 200 genes, targeted regions of interest. My panels and probes are designed and sequenced as per the technology. It could be Thermo Fisher, it could be Illumina, or now even the transcriptome. So earlier only DNA, now DNA, RNA, or only RNA also being sequenced. And the targeted NGS is the commonly practiced technology. Exome sequencing we typically reserve for not reserve, but typically used in the rare event or the hereditary cancers, where we use this technology, but lot in research settings, somatic exomes, I mean, also being spoken of. But now we are also seeing transcriptome applicability, particularly in ALL settings, and whole genome settings, again in rare event or wherever we don't get a diagnosis. So they are about to be there into the clinics. Not that every case we want to use it, but they are there. Earlier I used to say only targeted sequencing, but now I think the things are moving very fast, and we have most of these technologies available into the practice. How do we use it? So we have DNA extraction, we have RNA extraction. Prepare your libraries and then look for point mutations, as I mentioned, indels, copy number alterations, fusions, and even gene expression. So either I use DNA, RNA separately, or I mentioned total nucleic acid. I could use even total nucleic acid. So those are also being spoken of. In liquid biopsy settings, we commonly use cell-free TNA, and then process or for detection of all other things. a typical workflow, this is the basic workflow. I'm not going into the depth of it, so don't worry. This is just for you to understand that how does the workflow work in the lab. So you have extracted, you used your assay and you sequenced it. Whatever comes out of the sequence is usually FASTQ files. So till this FASTQ files, we call it something, we call it primary analysis. And we need FASTQ file. So from this FASTQ, you generate a BAM file or a VCF file, which has all the information. So, and this is done in comparison to the reference genome. We call it secondary analysis. So VCF has all the variants that we want to analyze. Now using this VCF file, we annotate it and interpret it as per our report. It is called as tertiary analysis. Why I'm specifically mentioning is because you might have different softwares which require FASTQ files. Some softwares require BAM files. For tertiary analysis, you require VCF files. So you need to be aware what files are there and what kind of interpretation you are looking at. All the equipments typically tend to follow the same. The wet lab processing, the primers, this could be different, but the overall workflow will remain the same. So you have now VCF file which has all the variants, mutations. My sample type being different. My sample type for a germline testing, as I mentioned, could be saliva, it could be skin biopsy, it could be peripheral blood without blast, post-induction sample for mutation in a hematology setting. So when I'm looking at germline mutations versus somatic mutations, so my reporting is slightly different, and there are a lot of guidelines available which we use for our clinical reporting. For my germline testing, we use ACMG or AMP criteria for reporting. For my somatic mutation reporting or classification, we have tier-based classification. So you will see a particular mutation as tier 1 or tier 2 if these are variants of potential clinical significance. And if tier 3 or tier 4 being labeled as benign or unknown clinical significance. So variants or changes where we don't know what to do in this setting. So this is another way just to share that the somatic variations are tier 1, tier 2, tier 3 and tier 4 or benign variants, and we report them into our clinical report. So the classification is different based on somatic or germline mutations. So one key factor or key thing that you all need to be aware, or we all need to be aware, is VAF, variant allele fraction. So I'm doing NGS. I need to be aware of VAF. What is VAF? So it represents the percentage of mutated DNA molecules relative to the total DNA output. Typically, the reads which show this mutation versus the total read generated in that area. And you will find that VAF reported in the end of the mutation. So these are usually in a report, NGS report, you should see these things. And you need to be aware of when you're reading that. So JAK2 is present at what variant allele fraction? And these additional findings, what change is present, what protein this change is there, at what location and what transcript ID, sometimes helps to, there are discrepancies or solving those discrepancies. These informations are very, very helpful and they should be mentioned in the report. The VAF is very, very critical to understand, and that this VAF has a lot of role in terms of at what percentage this is present, and this gives a lot of information, and it is dependent on not only on the size of clone, tumor heterogeneity, and also on the co-occurrence of other alterations. Suppose on one side there is a deletion, and on the other allele there is a point mutation at 25%. So you will have a VAF of around 75%. I'm simply saying like that. There are a lot of factors which influence and my understanding of VAF gives you a lot of information. So post-treatment VAF versus a diagnostic VAF, something close to 50% in the genes of interest, in the genes which are likely to be hereditary. They are helpful. So VAP is helpful to predict sometimes the germline mutations in the genes. Like in CEVP, if I'm getting a mutation of around 40-45%, I should rule out that whether it is germline or not. BRCA1, BRCA2 could be somatic, could be germline. RUNX1, it could be somatic, it could be germline. So these are the genes which are known to be hereditary, and any mutation or variation in the region of around, the guidelines say above 30%, but around 40 to 50% is what we consider for a routine thing. So these could be germline, and we need a separate buccal swab, an assay to prove that this is germline, or a post-induction sample. Another point is the depth and coverage, and so for germline variants we tend to sequence at around 100x. For somatic variants, more than 500x. So for liquid biopsy or rare event or NGS based MRD, the depth we are looking at is 20,000x and beyond. So more the sensitivity. And so sometimes, because this will generate a lot of data, and sometimes people tend to compromise on the data, and the sensitivities will be different. If we don't target in these regions, my sensitivities are likely to be low. So we need to be aware that this sequencing is done at what coverage, and this is some of the guidelines which are available to be able to do that. Last few slides, I'm not going to take much time. I think I've already taken much time, but these last few slides. So VAF, as I mentioned, since we are sequencing more and more, we are getting mutations at lower VAF, and now we know that the somatic mutations are present not only in disease and also in healthy processes. Like if I'm sequencing some of the healthy tissues, I will get some common mutations. As we know that in hematology, we have clonal hematopoiesis. Similarly, in other tissues also, there are low-level mutations detected. And since we are sequencing more and more, we need to be aware that not blindly labeling any low-level mutation into a disease process. So there is something called as CHIP in hematology. Now we are understanding more and more, and probably in going forward, you all might be looking at starting some CHIP clinics. I mean, this is what I think, like this is the way this is going. Like since we are sequencing more, we are detecting low level of mutations, and this typically these mutations in around 25 genes are described. We have low level mutations with a VAF of more than 2% being detected in the 25 genes which are described, and a lot of common genes are there. And we need to be aware that this could be normal clonopoietic hematopoiesis or normal aging process, and not labeling everything as a disease and labeling it. It has to fit into the clinical context, and then only take it into my clinical decision making. So we need to be aware, and VAF has an important role, more than 2%, typically less than 10% in the genes. This has been described. I am not going into the details of that, but awareness is very, very important. So coming to the end part, I shared with you a lot of PCR, a lot of assays, VAF, NGS, and I was mentioning that now we are looking at other high throughput technologies like whole genome, exome, and even transcriptome being coming into the clinical space, being able to provide you various other biomarkers which are there in clinically. One area where I think technology is coming into the hematology clinical practice is transcriptome, and for that now people have used RNA sequencing based classifier, and these are softwares available, ALL sorts, even MDALL. So different people, different groups using whole transcriptome, not targeted, whole transcriptome, and then using that software to be able to classify into the different 26 subtypes of ALL, because ALL is one where we are using karyotype, FISH, still, and a lot of newer subtypes are there, like PAX rearranged, PAX5 mutations. Then there are different fusions being described, ZNF384. So it was not into the research domain. It is coming into the clinical domain and also shown into by different. Again, this is showing transcriptome sequencing coming into an ALL clinical laboratory. So it works down to the clinical laboratory, not in research anymore. So I just, for the my last case is there. I just wanted to share this case. So this was a 10-year-old female case of pre-BALL, and we did what was required. Traditionally, we use cytogenetics and an NGS, and you see cytogenetics showing XY and add21 here, and NGS was showing mutation in the NRAS and KMT2D gene. So these were the two mutations. These are 200 gene panel showing mutations and fusion. So this is a typically what we do, karyotype, and if available FISH or an NGS to be able to pick up that. So here we used a transcriptome. So we have this now, just started this assay. This is a we call it a trans-CKLL. And you can see here, this was clearly showing that this is an intrachromosomal amplification of chromosome 21. So this is a subgroup which has a poor prognosis. So the gene expression profiling as well as the other were showing that this is an IM. So we were also showing that there are CNV alterations. So different more details are there, but I'm just cutting short that this was showing that this is an intrachromosomal amplification of chromosome 21, which was not picked up initially, but this was showing clearly that this is an intrachromosomal amplification, which you are not able to pick up on an NGS. Typically, we use FISH to be able to pick up. Hyperdiploidy, hypodiploidy, or chromosomal alterations typically don't pick up on an NGS, and we require either FISH, karyotype, or other MLPA technologies to pick up. So here we were showing that this is a IM, and we retrospectively processed that FISH for that, showing that this is present in that. So confirming whatever we were suspecting on that assay. So just thought of sharing that this is how the field is moving, and we all need to be aware of what is happening to this. A lot of exciting things are there. But coming to the end, and this I think is one of the most important, for everyone to know, what is the molecular analysis, what is the anticoagulant of choice that we use. So this is a common problem that we face, sending wrong samples, and then we have issues downstream. So EDTA is the anticoagulant of choice for molecular analysis, and heparin is the anticoagulant of choice for the cytogenetic analysis. And sending molecular analysis in heparin is an inhibitor of the PCRs. I showed a lot of assays affect those PCR, and heparin is one of the inhibitors. So you might end up in a scenario where your assay is not working just because you collected your sample in an heparin. Though sometimes we process it and it works, but still sending samples in the right anticoagulant is of paramount importance. And as I mentioned, the nucleic acid is a key factor to any of the processes that we do downstream, and we need to be aware of that. So with that, I come to the end of this, and I would like to thank you all for your patient listening. I hope this has been useful, and I'll be happy to take any questions if they're there. Thank you so much, sir.
Thank you, Neeraj, for that elaborate but very simplistic talk. You're a non-molecular pathologist also. It made a lot of sense. We have two questions in the chat box from clinicians. I'll put that to you first.
Yes, sir.
Mukul Agarwal wants to know, for exon 12 mutation polycythemia vera, you need blood sample or bone marrow sample?
So we can do with the peripheral blood sample, because there are abnormalities which are there. So we can use that sample, not a problem. We can use peripheral blood sample.
Dr. Shyam wants to know, please elaborate NGS for MRD monitoring in ALL. How does it differ from routine NGS at initial diagnosis?
So in an ALL setting, basically you are looking at IGH. So I want to target the receptors. So what I have to do is I have to characterize those clones in the initial settings. Either I use it by a real-time PCR based characterization, or I use an NGS based characterization. So I have to get those clones right, and then I have to monitor that those clones in the post-induction sample. Typically, this is real-time PCR is the assay to go where I mean, this has been typically used. NGS typically has been used, but not available in Indian settings as yet in those scenarios. So, but it gives you a little bit better sensitivity, maybe 1 log. So, but I typically, to me, flow is kind of the technique of choice, because it goes down to 10 raised to power minus 5, 6 setting, 10 raised to power minus 5. So, and it gives you within a 1 day. So why do you want to spend around 50,000-60,000 to pick up maybe 1 log more sensitivity and have 1 month the results coming after 3 to 4 weeks with a slightly difficult? But having said that, it has a little bit more sensitivity, technologically slightly more challenging. But I mean, that are some of my views in terms of, you can do that. It is not that it is not doable, but I feel need is slightly less.
Dr. Ranjit from CMC Ludhiana, where does Oxford nanopore technology in research or diagnostics?
So this is coming up in the space of long read, and it offers better sensitivity, but it is a costly technique. Still, there are the scales at what we are looking at is not feasible. We are looking at this technology into the diagnostic space, typically in an HLA setting now. I feel in the HLA setting it would be making sense initially, but going forward, if to the scale and if to the cost effectiveness we require, we might have that. Still, it is not there. And also in the fusion settings, you might have long read sequencing being practiced. You have RNA-seq, as I mentioned. So you use a long read RNA-seq and then use it for any of your further applications, primarily in the fusion and then even in the mutation. So the limiting factor is the availability, the issues with the Oxford, the mutation rate. There is a 5 to 10% mutation rate is there, and the cost. So those are the things which are limiting that, but it has got a very high potential. If we are able to work upon on these things, we can see that it is coming into the market or available.
Dr. Aditi Goyal wants to know, in CML, does molecular genetics help in selecting the use of a particular TKI beyond the conventional risk stratification?
I mean, we use that for our selecting the TKI based on the mutations that we propose TKI, that based on different types of mutations, and their sensitivities are known. So we use that very commonly in the kinase domain mutation analysis assay. Yes.
I may just add, Dr. Aditi, that we don't do this at baseline. We do it when a particular TKI fails. She has another question: cases where there is a borderline BCR-able transcript level, very low copies. How do you differentiate between residual disease and a false positive?
That is a challenge area. It is a challenge. Post-treatment, getting low level values. So it is difficult area. We do get contamination issues. So having a subsequent sample and comparing with the rest of the samples, if all of your samples are showing that low level of positivity, around 50 reads, 100 reads, so you need to be a little careful when you are reporting. A repeat testing, or if it is not matching into the clinical profile, suddenly values were detected, zero values were detected in a case which had 2% or 5% values and not matching clinically. So that is a time when we kind of correlate, but it is a troublesome area. So that is why providing history, when you are sending your samples, or that if that we have that history, many times we correlate and even discuss that.
Again, Dr. Aditi, the recommendation is repeat it after 1 month, as Dr. Neeraj said, and the both should be consistent with each other. Dr. Nikhil wants to know, sorry, not Dr. Nikhil, Dr. Hita wants to know, for somatic mutation in AML and ALL, if there are blasts in the peripheral blood, is the peripheral blood sample as good as bone marrow sample? How many blasts should be there in the peripheral blood for the mutations to be picked up?
So again, there is no cutoff available. The more percentage of blasts are available, the better. Typically, 10 to 20% blasts are sufficient for processing. Again, that depends upon the technique that you want to use. If I'm using an NGS, so that amount of blasts are sufficient. But if I'm using a Sanger sequencing, so then those blasts may not be very sufficient. So, so you can have 10 to 20% blasts and then use that sample for downstream NGS. If those blasts, otherwise you have to have a bone marrow sample for that, because it gives you, even allows you to pick up, to add karyotype assay as well. On a peripheral blood sample, your karyotype may not work well. So that is why doing a bone marrow is always preferable in those settings.
Rishi Bansal wants to know, kindly elaborate the role of NGS-based MRD for AML as against flow-based MRD.
So as I mentioned, I think for ALL also, for ALL, it is very nick and corner. It is very parallel to whatever you get on flow. For in AML settings, the NGS-based MRD has a role, because the flow picks up around 70% of the cases, and the NGS picks up other 20-30% of the cases. So having a complementary flow plus an NGS MRD is helpful. But having said that, a right assay to be able to pick up all the variations or mutations or fusions down to the sensitivity that we need is quite expensive. So better sense would be having a digital PCR based sensitivity for those fusions or mutations. So this is a touch and go, but those are complementary, and we can have NGS based MRD added to the flow settings. There will be a cost. It will allow you to pick up around 20% cases more.
Thank you. Dr. Vinayak Chekodi from Pune wants to know, what are wild type genes and mutations?
Very good.
So wild type means those are negative. When I have got a sequence or done an assay, so wild type means it is a negative. So whatever sequence normally is there, it is there. So that is meaning wild type. If mutation means there is a change in the variation. So if a change is there, change means T2A. So any base pair is changed to the other one, leading to the change of the protein there. So that is a mutation or a change. T2A, amino acid change or a base pair change. So that is a mutation. I hope this is clear. Wild type means it is negative. Mutation means a change. It could be a deletion, point mutation, insertion. Everything would come under the scope of mutation. It could be a large deletion. It could be a small deletion. It could be an indel insertion as well. So that is all a part of mutation.
We have 5 questions from the participants here. So starting with Dr. Rajat, your question, Rajat.
Sir, Neeraj, what a fantastic talk. I was glued to the system. So my question to you is that, of course, the transcriptome analysis should be a standard for all ALL children. What about AMLs? Is there something now that in ALL we see 26 variants, AML probably even more. So do you have something for AMLs which will pick up more targetable lesions?
So why I mentioned ALL was because ALL, there are challenges. So one assay being able to pick up and the availability of the right assay. So AML, it is more or less defined. We are able to get the mutations, we are able to get the fusions. So more or less they are defined. For an ALL, still you sometimes want to use fish, karyotype, you want to use microarray or GEP, whatever so there is where a single technology coming into the practice. Having said that, transcriptome has been used in AML settings, and there is an NEGM paper as well, which is used only transcriptome based assay to be able to pick up all different subtypes and even more. The problematic areas being typically when you use RNA, getting the mutations, low-level mutations, like 5 to 10% of those mutations accurately from that RNA is as a problematic area when you use whole transcriptome for your mutation sensitivity. So around 90% of the time, 95% of the times you are able to pick up the problematic areas being these mutations. For an ALL, mostly fusion, so it doesn't matter much. So we are able to pick up all those things because that is where your interest is. For an AML, mutations being your area of interest, you want to miss out those 5-10% cases is the discussion or the thought process in that. But it has been tried and it is available. A lot of data is available, but still not into the clinical space as yet. You have ample things available for your treatment and diagnostic purposes, is what I feel.
Okay, thank you.
Thank you, Rajat. I'll pick up 2 more questions from the audience. Dr. Shyam wants to know, how do you interpret unexpected mutations detected by NGS when you are doing it for a different purpose? For example, NGS is done for immunodeficiency, and you find some mutation suggestive of TTP or Glanzmann's.
Yeah, so whenever we do an NGS in these settings, we have to have a phenotype. And if we, what mutations we get, we correlate with the phenotype that is given. And if that matches is what we report initially, that variant has been or mutation has been detected as per the phenotype or as per the clinical indication. So for any such things which we detect, those are incidental findings, and we report those as incidental. We discuss with that. Many a times there are the TRF mentions that whether you want those incidental findings to be reported or not. So based on the clinicians or somebody who's requesting that whether those incidental findings are to be reported or not, those are reported. But we tend to include that. But again, based on the clinical clinician, if they want that those to be reported, we will report, otherwise not.
Dr. Apeksha wants to know, why are some mutations picked up by FISH and not by NGS?
I don't know what the exact question is. Mutations typically are picked up by sequencing or other assays. We use FISH for whatever question you are asking. My question is whether BCR-ABL is present or not, it will pick up. PML is there or not. So that is the question that FISH tend to answer. So if you are asking those questions and using that particular probe, or then you are able to pick up, use FISH. I am not sure that are there any assays which say that only FISH is going to be. Yes, there are like IAMP is one such assay where FISH is going to be picked up. But typically other things NGS picks up in mutation settings. I hope this is clear.
Dr. Shyam has one more question. Is NGS MRD always superior to flow MRD for acute leukemia?
No, not like that. So these are both depending upon the disease, as I mentioned. ALL settings, those are almost complementary, but we can still see cases picked up on an NGS and not picked up on flow, or picked up on flow and not picked up on NGS. There will be cases like that, but the percentage is very low. But as opposed to ALL, AML settings, there could be a lot of cases where either a technique which is picked up. So more number of cases are picked up on NGS and not picked up on flow, and picked up on flow and not picked up on NGS. So there are cases, and I think Nikhil has a paper published in this area where around 20% cases are like that. And also this is dependent upon the expertise available to be able to analyze and interpret that data. Not many experts are available in this space, so leading to a lot of discrepancies. You might find more discrepancies as we see the level of expertise dropping down. So having two complementary technologies in that space would be helpful.
Dr. Babita, your question.
Thank you very much, sir. First of all, good afternoon, everyone, especially to Dr. M.B. Agarwal and Dr. Shashi Bansal and Dr. Neeraj Arora for having the very beautiful talk. Sir, I am myself MD Pathology working in Delhi Max. I met Dr. Neeraj in Bombay this year in the month of January when the conference of MHG was held. Perhaps you remember, sir.
Yes.
And sir, today I'm thankful to Dr. M.B. Agarwal for which the purpose I'm part of this team. And sir, for the thanks Dr. Neeraj for having a wonderful, very explicit talk and a concept making talk, sir, on this topic. We are, I'm of the opinion, sir, that with these types of talks, we can make our labs equal to the standard of the international level. And sir, this is, I'm again of the opinion that ABC and the basics of the any topic, ABCD, any topic maybe, can be a step ladder, can be a challenge, can be a central axis around which we can revolve. And this is a very concept making talk. Sir, I have two queries. First of all, sir, in start of the presentation, you just said about the mutations that I mean it is a change in the mutations, change in the primer, and change in the purine to pyrimidine. It can be pyrimidine to pyrimidine also.
Yeah, it can be there. So that is, so I was just mentioning that though clinically, I mean, I don't consider that too much into my clinical practice, but yes, any changes which are affecting that, it could be in that way. That is not a thing. It can happen.
Then my second query is that, sir, that in carrying forward the same question by the earlier asked, that what is the difference between the wild type and the normal? Sir, every wild type, every normal, we may not call it as a wild type. So what we have not much understood about the wild type vis-a-vis the normal.
I think I have not made it clear. Typically, when it is normal, we use a terminology called wild type, and we have to be a little more clear when we describe that wild type means normal. That I think we have to be very clear now. When I realize that probably I'm not this side, and maybe my clinical colleagues as well, somebody who's reading that report may not interpret in the way that it is intended. Usually we write wild type, meaning there is no change, or it is similar to what is described. Meaning this is normal. That is what we tend to communicate.
Sir, again, just a comment on, we were so blessed to have your talk today. Just a comment on that, I mean, about that this thing is if we want to achieve, if we want to go to the XYZ, we must start from ABC. That is the carry home message. And if we achieve XYZ, we should not forget about the ABC. So that is the carry home message I always tell to my students. Sir, then at the end, we just want to have your email ID, sir.
Sure,
Dr. Neeraj.
I will. Sure.
Thank you very much, sir.
Thank you, Dr. Neha.
Good afternoon, dear all. And same as Bhavita Madam, I had the pleasure of listening to Sir in the January conference. So I have just one clinical question. We had a 50-year-old gentleman with just complaints of thrombocytosis and no other relevant complaints. One episode, thromboembolic episode, some 2-3 years back. But he did a MPN panel. So the MPN panel was positive for SF3B1 as a prognostic marker and MPL as a diagnostic marker, and both having a VAF of around SF3B1 with a VAF of 31% and MPL with a VAF of 29%. So I just wanted to understand when this kind of prognostic and diagnostic markers are being labeled, what is the kind of relation that we have with VAF? So for example, in this case, the VAF is pretty much similar for both. But then I believe in what category to place this patient, either into MDS or probably an MPN, wherein SF3B1 is just an indicator of increased thromboembolic episodes. So we were in a catch situation here, so I would love to have some inputs from Neeraj sir regarding this.
So when it would require the clinical context as well, as I mentioned. So any mutation, any change without the clinical context, we should not be placing them into the category. So again, you would want to look at what, as you mentioned, TLC, DLC, platelet count is high. And typically you are looking at an MPN kind of a scenario, and there you are getting an MPL mutation. Then you would have a consideration of ET. Again, that would you want to consider that diagnosis. but VAF is critical here. Many a times the MPL mutations are also germline, and the 515. I forgot the mutation. Typically, those are the hotspots in this region, and many of them are germline. So you have to rule out that implications as well, and specifically with MPL. Otherwise, like SF3B1, in this scenario, I won't be able to comment what is the role of SF3B1 typically in the scenario of an ET. In terms of treating sense, I don't think that is sir can add on to that. How does that affect the clinical treating perspective? But diagnostic perspective, it is not too much significant. But I would still like to evaluate that fully. Peripheral blood, CBC, what is those things are like. Then take a call on that.
Okay, unfortunately, we did not have a marrow procedure done for this patient. Peripheral smear wasn't too significant, just thrombocytosis all around, and not much literature available, regarding this. So I could only comprehend the fact that SF3B1 associated with a lot of thromboembolic episodes when associated with MPL. But then I really was confused regarding, the VAF being similar and one being labeled as just a prognostic marker and one being labeled as a diagnostic marker. So absolutely, I love to hear inputs from Agarwal sir regarding this.
Okay, you have thrombocytosis. You have had a clinical thromboembolic episode already. So clinical picture is that of essential thrombocythemia. And in that MPL mutation, to a clinician will look like a somatic mutation. Diagnostic, so it's a driver mutation over here, unless you pick up that this was germline and not somatic. That's one part of it. Non-driver mutation like SF3B1 in this setup has one significance, especially when you're not done bone marrow, is the RARS with thrombocytosis, refractory anemia with ring sideroblast with thrombocytosis is one entity which is associated with SF3B1. And then we have a specific treatment over there in the form of luspatercept. But then those patients very rarely will present with pure thrombocytosis. So in the absence of bone marrow, in the absence of seeing the ring sideroblast, etc., I will just call it ET, MPL as the driver mutation, SF3B1 as the non-driver mutation, act only on the ET for the treatment, will not label him as MDS at all.
Sure, lovely. Thank you. Thank you, sir, for your insights. Thank you so much.
Take one question from the audience again. Dr. Mukul Agarwal. Sometimes the reports from PCR and NGS are not concordant. Is it because of the sensitivity of the tests or any other reasons are possible?
So there could be many reasons. Technique sensitivity is one of the reasons. The sampling is other. You might have sampled. This is a common scenario that we see, that one assay is sent from a peripheral blood and the other assay is sent from a bone marrow, and this leads to discrepancies. But typically the assay sensitivity is one of the major reasons to be able to detect that. And sensitivity of an NGS is slightly more. We are able to detect go down to 1 to 5%. Again, this would be in contextual, what context you are talking. There could be other scenarios as well, being picked up on an PCR and not picked up on NGS, especially FLT3 ITDs also. When those are, so both ways are there. So we need to be aware and in that sense.
Sure. Dr. Kakunje Manasa, your question.
Yeah, good afternoon, sir. Thank you, Neeraj sir, for the talk, and thank you, Agarwal sir, for the opportunity to be a part of the discussion. Sir, being a clinician, I have a very practical question. Like, is skin fibroblast culture karyotyping being done in any lab right now?
Not clinically done, so that is why I did not want to focus. But that is what the recommendation is when you want to. Not that I am aware that it is routinely available, but typically we don't, not available clinically, but CMC does that. That is what, again, I am aware because we used to do earlier for some of the cases.
because so my question is like to differentiate between germline and somatic. You have explained everything, but then we had faced a practical problem when a child with clinically Downs, but not confirmed previously, but diagnosed to have leukemia ALL. And on karyotyping, we found the karyotyping we sent from the bone marrow for the for the cytogenetics. The patient was found to have trisomy 21. So in this situation, without a previous karyotyping on the patient, it was difficult for us to consider that as a
Whether it is
like
constitutional or whether it is somatic, like that.
germline or something. And we did not find at that moment, we couldn't find any labs doing the skin fibroblast as well. So what do we do in such a scenario?
So I mean, follow-up sample later on, maybe when there are no blasts or anything like that. So that would be again be helpful.
So here the patient, like it becomes very important because patient with Downs giving the chemotherapy, whether the dose modification it needs to be done or not, that will have to be decided initially. So it becomes practically difficult to send the sample at later stage. So
Can you
So-
do a buccal swab? Sorry, Neeraj, I'm just interrupting. Buccal swab, will that give you the answer?
okay, so
Yeah.
just a karyotyping on buccal swab would be on. Hello.
Yeah, I mean, I'm not too aware. Skin fiber, I can check with my team, cytogenetic team regarding the availability. I don't think it is available. But what additionally can be done, I can discuss that. I am not fully aware.
Okay. Thank you, sir. Thank you.
What about Raj's suggestion of buccal swab?
Yeah, I mean, again, I don't know whether you will be able to do karyotyping on that or not. So I'm not too sure.
Okay.
Because karyotyping, you need cells for your culture.
Okay.
Thank you, sir.
As a clinician, I can just say that clinically, if it is Down syndrome, it is Down syndrome. It is so, I mean, Rajat is there as a pediatrician, but when clinically you are seeing that there's a Down syndrome, it is very difficult to challenge it in this setting. And I would always then use the protocol of chemotherapy like Down syndrome. Avinash, your question.
Yes, sir. Sir, first of all, just we discussed about the SF3B1. So I think a recent paper of Taferi said that there can be SF3B1 mutation with MPL in ET and others, and they prognosticated with some certain risk strategy scoring status also. So rather than ASXL1, SF3B1 has its role, and I think so around in 5% cases you can get SF3B1 in ET also. And sir, what about if we are getting low BCR ABL copies? So why we cannot go for ddPCR and others? Or we can think about the ABL copies if they are in lakhs and the BCR ABL copies are less and ABL copies are sufficient. So and there is no log progression. So I don't think so. There should be any confusion in that.
Regarding, I mean, you're saying use of ddPCR versus real time is what you're mentioning?
Sir, the question came now regarding that if you are getting low level of BCR ABL, then what to do, how to confirm whether they are truly low or no. So in that, I think ddPCR can help. And again, if on real-time PCR, if you are getting a particular number of ABL copies, you are getting 3 lakhs, more than 3 lakh copies. So according to that criteria, if you are getting 50 copies, then also it will fall into your MMR or DMR group.
Sometimes false positive is what.
Yeah. So we can rule out by ddPCR also, right?
Yeah, I mean, you can use ddPCR, even you can use real-time PCR. The problem is whether it is false positive. So if there is a contamination or if something is there, so even if you use, so basically using other assay would help you, certainly help you confirm either real-time or a ddPCR, whichever assays you use.
DD. So no need to go again and wait for 1 month. We can simultaneously, if we have RNA, and then we can go ahead.
You can use that.
And
certainly,
sir,
as Sir also mentioned, repeat testing is also helpful in those scenarios.
And one more question was floating around the MRD, what flow or other thing. So I think the 22 guidelines were very straight for AML. You should go for both. If both came negative, then the prognosis is very good. If one came negative, one positive, then it's sort of intermediate. And there is a lot of false positivity and negativity. And you, as you rightly said, that it's about the expertise. So the MRD and AML, it's like a tough job. And you made it very simple for everybody, that all these things like NGS but we know that all these things are not that simple, and they have the things. So thank you, sir,
Challenges.
for a beautiful and such a beautiful talk. But the thing is, like ALL, MRD and MRD, if you are working in resource constraint setting, you have no time, then the flow is the best thing for go. And if you're getting a proper experienced person and doing this, you can believe on that and you can go ahead with that only. Thank you, sir. Thank you, Agrawal sir, for giving me the opportunity to be here on this stage as a discussant.
Thank you. One more question from the audience. Dr. Vinayak Chokodi wants to know, germline mutation, does it mean it is heritable?
Yeah, I mean, germline is heritable means you want to confirm first it is germline or not. Then it is going to be passed on to the offsprings for sure. So that is why it is there. And we want to rule out whether other family members are affected or not. So that is what, yes.
Yes, the answer is yes. Okay. Dr. Nishat.
Good afternoon, sir. So, sir, wherever possible, do you recommend that bone marrow is a better sample for, molecular test as compared to the peripheral blood? And in case, if you have a strong clinical suspicion and especially in myeloproliferative disorders, if your peripheral blood NGS is negative or peripheral blood PCR is positive, do you recommend repeating it from the bone marrow sample?
Like, so you say that if your peripheral blood sample is negative for PCR and then using NGS, right?
Suppose the peripheral blood sample is negative for either PCR or NGS, do you recommend it doing from the bone marrow sample?
Not really. In an MPN setting, it doesn't affect that. If usually peripheral blood is sufficient sample to be able to pick up all the abnormalities. I have not seen MPN cases being picked up on a bone marrow and negative on a peripheral blood. I think peripheral blood is a good enough sample in this scenario.
Sir, I just wanted to mention, I had a patient who, had a clinical suspicion of ET. She had a thrombosis and her peripheral blood PCR was negative for, MPN reflex panel was negative. And that time a marrow wasn't done, but subsequently 2-3 years later when a marrow was done and the NGS was done from the marrow, she turned out to be JAK2 positive, and that is why I'm raising this question.
Maybe initially sample. So I think this is again important to highlight that molecular labs, once the DNA is extracted and if it is not labeled properly, there could be sample mix-up issues there, and these are true issues because we handle so many samples are there. So maybe a repeat sample testing also is helpful, and I am true to admit there could be sampling issues in the molecular lab because it is very difficult to, once the sample is labeled into particular thing, I'll be using the same sample. So if there is a discrepancy or if there is not matching into the clinical profile, I always tend to have a repeat sample retesting additional sample rather than using the sample or a re-extraction samples and then test it out. So this we tend to use in the practice, but I don't know per se peripheral blood versus bone marrow. I think it should be able to pick up. Maybe you can try now as well, sending a peripheral blood sample and see that this is getting picked up.
Another case I would like to mention here. I was monitoring a patient of 821 AML by AML1 ETO PCR. Samples from peripheral blood used to be negative, but simultaneously the samples from the bone marrow used to be positive, and eventually this patient relapsed, frank relapsed. Then I did a haploidentical stem cell transplant for that patient after a salvage. And now, she's in remission. But since previously she had a bone marrow, bone marrow used to be positive for PCR, I have, the patient also insists that to monitor the 821 by bone marrow. So we are doing the bone marrows every 3 months or every 6 months for this patient.
scenario, it might be feasible or possible to do that. But again, depending upon the mutation type, like for an NPM or peripheral blood monitoring would be sufficient. So that way, it might slightly differ peripheral blood versus bone marrow, what we need to do. In an MRD setting would be slightly different. But still peripheral blood is there for NPM scenarios.
Thank you, sir.
Thank you.
Dr. Bharat?
Good morning, sir. And as a medical oncologist, I still like to attend your seminars because of the quality of the content and exceptional authors and speakers you invite. So my question is, one is a question and one is a suggestion. I'll start with the question first. In NGS of solid tumors, many a times when we do full exome analysis or a transcriptome analysis, there are certain mutations which are kind of specific for hematological malignancies, for example, BCL2. So many a times in solid cancers, when an NGS report comes, we find certain mutation which is of hematological origin. And the review of literature does not show strong presence of such mutations in solid malignancies. For example, as I said, BCL2 or CDK in colon cancer. So how to respond to that?
I think I shared that slide purposely for that scenario only. I showed the CHIP scenario or somatic mutations being detected in normal diseases as well. And there are around a list of 25 genes. I can share that 25 genes, common genes, which are mentioned in hematology. And those are common like SF3B1, BRAF, those mutations, which are normally present. And I also felt that solid tumor testing, these were not very clearly described, even JAK2, low level of JAK2 being detected in those samples, which we tend to ignore earlier. So in maybe lung cancer, sometimes you get 2% JAK2. I mean, you would tend to ignore that scenario. But after having read this paper, you don't want to ignore and maybe document those cases and see whether what changes or what things are, they are there and need to be correlated with the normal description of those mutations in those normal tissues, particularly maybe in a liver tissue, I'm getting low level of mutations, or in another tissue, whether those are described in those settings. So that is what I tend to do now, since I do both solid and liquid, but having meaning for those. Again, in the clinical context, only you would be valuing that. Other than, I won't value any specific mutation other than the specific clinical scenario. And here also VAF is important. What is the VAF at what these mutations are being detected? Whether it is less than 10% or it is in a significant proportion. So if I am getting in a lung cancer, maybe a hematological mutation, typically described at a VAF of 20-30%. So then I would tend to reach out to my colleagues, clinical, oncologists, and then discuss with them what does, since I'm getting after confirmation. So this could be false positive, repeat, a lot of things can be there. After ruling out that, reaching out to them, whether this makes sense in the clinical scenario that these mutations have been detected, and then only reporting those kind of mutations. Blindly reporting, certainly at a level of less than 10%, is kind of more part of a normal chip clone. So that is what I kind of do that now.
Yeah. So since we also have other participants, there are certain AI tools which is, of course, it's evolving. Based on the NGS reports, it will determine the approximate cellular pathways, and will also do the review of literature and give approximate things that these out-of-box mutations can help a patient or not. These are a generative AI, so result may be a generation of their imagination. But yes, that is also possible. Now, second thing is my suggestion. Till 2002, I never entered any of the wet labs or the laboratories I was a pure clinician till that time, and I face the same problems. When the report comes and there is something written, I have no clue what is the methodology, the technology, the machines, and etc. And in my first exposure to laboratory, what happened in 2017, and I felt so insulted because I didn't know any of this methodology. I just walked out from that lab. It happened in University of Vienna. So anyhow, working on this disrespect to myself that I don't know anything. In 2002, I actually went to a basic science lab where I worked on cell culture and incubators and all these machines and all. So if I may request, if you can share approximate YouTube links of the videos explaining how this machine, I'm not saying animation, how the machine works, but the actual methodology which is followed in the lab. For example, a blood sample comes and what you do the next, what buffer you add, and this may not be your own videos, but a list of the YouTube videos which can give us clinicians, okay, this is what happens with the blood sent to the lab, and then how much amount of quantity is gone into the machine. So that will be very helpful because I stand at both the points. A clinician who has never worked in lab, and post 22, I worked in lab, and I know what was the gap between my knowledge.
So, I mean, thank you so much for that suggestion. So speaking to someone who has not worked in lab, sometimes it's very, very challenging and
Very challenging.
Very challenging. So I try to keep some pictures, photos of what we talk, and try to make sense in terms of what is there. But covering everything in a single 1 hour lecture is very, very challenging. We can have.
That is why I don't want it to be
No,
a
I understand.
presentation.
So we can take that suggestion. We have a group. We have a Molecular Pathology Association of India, and I happen to be the secretary of that. We can take that suggestion, and we have a website as well where we can upload this for everyone's, everyone who wants to reach out, or this is a good suggestion. We can see how we can upload in that website. So we, as an MPI, there is a website, and on that website we can upload few videos, and there are a lot of videos available. So we can try doing that.
Thank you. Because I always used to the MTT assay. What is the MTT assay? After working in the lab, I realized, oh, it's a simple photogravure analysis. Simple photogravure analysis. So that's why.
Thank you.
Thank you.
I have a little comment, and I'll ask your opinion also about JAK2.
Sir.
JAK2 mutation can be present in normal population. When JAK2 mutation is found, one cannot label the patient as MPL. Even in the patient of erythrocytosis, to call that patient as polycythemia vera, you require some more documentation, like WBC, platelets, spleen, bone marrow hypercellularity, etc., etc. So that is one kind of mutation which can be an innocent mutation, and one has to be very careful in labeling the patient, especially if the clinical picture is not total. I've seen people avoiding bone marrow examination in such patient, which can be a disaster between erythrocytosis and polycythemia vera, just based on JAK2, because once JAK2 is positive, hemoglobin is high, polycythemia vera, it's not correct. Can be problem in follow-up. Any comment on that from your side, Neeraj?
Sir, I mean, so there is a lot of literature coming into, sir, in specifically this scenario, normal scenario, and I was surprised to see that there are TET2 ASLX mutations being described in normal neutrophils as well, and this is correlated with the cardiac activities. So if I have a particular set of mutations, I am more prone to thromboembolism. That kind of correlation is coming into, and these are normal low level of clones which are there, not that very high level of clones. So these mutations kind of affect the normal function of these leukocytes, and these leukocytes behave abnormally. They lose their normal, or they alter, I won't say lose their, they behave abnormally and then behave in different ways, and this sort of feature leading to more thromboembolic, more prone to immune features, that kind of literature is coming into what I saw. That I didn't share that particular paper has more in detail, and that is why awareness about this chip and these low level clones is very important for us to highlight that. And that is what I was reading about, that people are describing chip clinic, because how to understand those low level mutations and what is the meaning of that low level mutations, we all need to be on the same page, which we are not at that. We tend to ignore that, but knowing that and then seeing that this is significant and then trying to correlate with the clinical picture is what we will have to do. You have rightly pointed out regarding JAK2. And when we were earlier also reporting in some of our NGS solid tumors specifically, we used to get those, we still continue to get that, but we don't value that and we tend to ignore that. But now I realize there are those mutations, and this might have an impact, or of course more knowledge is required in this setting still. But it will be there, is what I believe. Specifically in cardiovascular scenarios, those correlations with those it is there. I did not show that slide, but it is there.
Thank you. One more question from the audience. Dr. Shah wants to know which labs are doing NGS MRD in India.
So there are many labs, I think, doing NGS MRD now. Not available in the ALL settings. I think maybe 1 or 2 labs are offering those tests in the ALL settings, but AML settings, there are few labs which are doing NGS-based MRDs.
Dr. Neha, your question.
Yes, sir. So this is in continuation with the clonal hematopoiesis concept that sir was talking about. So the DNMT3A, XSL1, TET2, all of these mutations, they increase the propensity of the background of chronic inflammation, which in turn leads to increased chances of atherosclerosis and atherombolism. So we have 2 projects ongoing. Thankfully, we have grants for that. We are yet to begin it. One in cardiovascular and one in chronic kidney disorders as well. So my question for sir is, what would you suggest? Would peripheral blood be preferred, or should a bone marrow sample be preferred for identifying chip mutations with a lower VAF as opposed to the diagnostic markers? And I mean, even if we do a peripheral blood, maybe sequencing studies on a peripheral blood, what should be kept in mind when we are looking at these kind of chip mutations with very lower VAFs?
It's a difficult question to know what to pick up. So ideally you would want to evaluate both. You can see peripheral blood and see bone marrow as well. Whether you are matching those, whatever is seen in peripheral blood, and how's the difference in the bone marrow. So if you are taking up as a project, if feasible, you doing bone marrow is again feasibility concerns would be there. Very difficult. So ideally most people will tend to do a peripheral blood evaluation, but if feasible, doing both would be very helpful, even in a less number of people as well, because these are normal people and you want to do a bone marrow for them, it is very difficult. So feasibility wise, I think peripheral blood would be there, and whatever you are doing, I think 2 times doing that assay
duplicate
assays would be to reconfirm whatever you are getting.
Yes. Okay, great. Thank you. Thank you, sir. Thank you.
I think that's the last question. So, Neeraj, thank you very much. You are with us for the last 2.5 hours. Wonderful talk and then wonderful QA session. I think everybody's enriched. The practice and the help to the patients will be different from tomorrow. Thank you very much and have a wonderful rest of Sunday at home or your workplace. Okay.
So much, sir. Thank you so much.
Sir, I can ask one last question, last query.
Please go ahead.
Sir, Dr. Neeraj, as you have explained every technology in a very beautiful way, but my query is that is there any one-stop show that whether we can say that this is the 99% and the more than that can be picked up by that? What is your comment on this, sir?
Ma'am, usually there is no single assay as yet. Each assay has their own limitations. I mean, if from my perspective you're asking what assay, so then whole genome possibly could be there, but it comes with a certain cost, baggages, lot of issues are there. So I think in our scenario, it has to be in the clinical context in what we are doing. If I want to know fish for PML Rara, maybe PML Rara I want to pick up, maybe fish is the best technology. It allows you to pick up within 2 hours or 3 hours. So depending upon that, you are able to pick up the technology and use in that scenario. Though I would say whole genome would be the best technology to pick up. But in the required clinical practice or the need, my need is to be able to pick up now. Otherwise, I'm losing the patient. So in that context, my choice of technology would be there. What I want to use.
Sir, in context of that NGS vis-a-vis fish, and how can we compare these two technologies, and how can we?
NGS allows you to pick up almost everything now. Almost everything can be picked up using technologies like NGS. But again, my cost, my TAT, those, whether those panels are covered, all those matter when I'm picking up my technology. So if my clinician is as good as Dr. Agarwal, sir, probably maybe a small Jack 2 will be sufficient because he knows the clinical context. But if you have a scenario, people like us want to, pick up, so just want to pick up everything. So we want to use maybe whole genome, So my choice would depend upon everything.
Thank you very much, sir.
Thank you. Hand over to Prashant Agarwal for the vote of thanks. Prashant.
Yeah, thank you very much, sir. It was indeed a very excellent presentation. You made the subject so easy and understandable. It was very interesting and intriguing. Once again, I thank Dr. M.B. Agrawal, sir, for giving us the opportunity, and the kind of questions everyone has asked was amazing. So it was a very wonderful session indeed. Thank you, sir.
Thank you.
Bye-bye.
Thank you, sir. Bye, sir.
Bye.